Today we're going to cover the scientific method and review what you've already learned in both your other classes in 6th and 7th grade. How do scientists gather information in order to answer questions? Take a moment, write your answers down, and we'll get back to it in a second. So hopefully you got a chance to write down your answers. So basically, scientists actually make a lot of observations using their five senses, using background resources such as primary resources, which usually include scientific journals, but not always. Wikipedia is not the number one source. You guys should know that by now. Making logical inferences from observations. Inferences are judgments or conclusions reached on the basis of the evidence or the observations you already had. In addition, scientists conduct experiments using the scientific method, which we're going to cover in a couple slides. Additionally, scientists draw conclusions based on their experimental evidence and the data they collected as they analyze and examine all the information. What are the forbidden rules of a scientist? Take a moment, jot some stuff down, and we'll get back to it in a second. Scientists definitely don't make conclusions without collecting data or performing an experiment. Usually, if a scientist or even a doctor, if they're going to say something, they always have some facts or factual information to back up what they're saying and have a reason behind it. Also, scientists don't base conclusions off opinions or biased data. Biased means that you're thinking about something and you want it to go a certain way. So you're definitely going to sway your results or make it seem like something's better than it is, or you're kind of like lying. You're giving your opinion and trying to sway everyone. Also, performing poorly controlled experiments is a no-go. We're going to explore what a controlled experiment and a well thought out experiment looks like. Based on the couple classes that we've already had, how would you define the scientific method? Take a moment, write it down, and we'll get to it. Simple definition for the scientific method are common steps scientists use to gather information and answer questions about the world around us. Very simple. You guys use it every single day, from when you wake up and you lost something, or trying to figure out a school problem in math class, or just like where you left your lunchbox in class, or where you lost your sweatshirt. Additionally, basic steps we've already covered are identifying the problem that you want to solve by the end of the scientific method. You're going to form a hypothesis, which is usually in the form of an if-then statement. We've given some examples in class, but we're going to go over one at the end of this whole entire PowerPoint. In addition, you're going to create some form of experiment that needs to have a control group, one variable being tested, and you're going to collect data based on your experiment, where you're going to analyze the data. After analyzing the data, you're going to formulate some sort of conclusion. This is where your background knowledge and your research is really going to come into play, because now you're going to be able to use everything you read about, everything you found, and you're going to try and explain why you got your conclusions, why the data came in the way it did, and any possible mix-ups or problems that could have happened. Now, your first step in the scientific method is coming up with some sort of problem question. It's always based on some previous knowledge or observations you've made in the past, and when you create your problem question, it should be something testable. What are your questions you're trying to answer? And you should be able to create some sort of experiment based on your question. Doesn't have to have a direction yet of what you want to test. It's just a general question. Why do plants need sunlight to grow? Why do plants need water to grow? Something along those lines. Secondly, your hypothesis. That's where you're going to take your question and you're going to give it some sort of direction based on the research that you've already conducted. So it's a predicted answer to your problem question. It uses that background knowledge and it follows an if-then statement. For example, if plants require water to grow, then 2 cups of water a day will make my plant grow bigger. Or if plants require sunlight to grow, the more sunlight I give my plant, the bigger it'll grow. After you figured out your problem question and wrote your hypothesis of where you want to go, now you have to set up your experiment. How am I going to test what I just said is going to happen or made a statement about what is going to happen? So take a moment and answer how many groups should be tested in an experimental group. At least how many groups? Hopefully you got a chance to at least answer that there should be at least 2 groups. There should always be a control group used for comparison. And there's always at least one experimental group that you're actually giving the variable or changing the variable in order to collect some form of data. In our example with plants and sunlight, our control group may be a standard of 8 hours of light for that plant. And then your experimental group, one group may get 6 hours, one group may get 4 hours, one group may get 10 or 12 hours. You can have multiple experimental groups, but you should always have at least one control group and one experimental group. Take a moment, and I'd like you to try and come up with a difference between what a control group and an experimental group is. Hopefully you jotted down some answers, and we can go over. Your control group is your default group. Just like you have your computer default settings or your cell phone default settings, it doesn't get any special treatment. It's not getting that variable that you're trying to test and see how it affects your experiment. It serves as a point of comparison. You always need something to look back at and understand, oh, wow, this variable caused the change in my experiment. This is why the data is different in my experimental group than my control group. Now, the experimental group is going to receive that treatment. It only has one difference. If you give more than one difference or more than one variable, you're going to mess your results up. You're not going to know which caused the change or what is making your plant grow bigger or smaller. And you know that if you're testing one variable, that difference in your experimental group is just due to that one variable. So let's look at an example. The problem question here is, does water have an effect on seeds growth? So the hypothesis is if plants require water to grow, then seeds will require water to grow as well. So what you're looking at is on this left-hand side, you have everything being exactly identical, two exactly the same pots, same amount of dirt, same amount of seeds, same type of seeds, same amount of sunlight. As you can notice, I think it's trying to show you that it's on a windowsill. So if you could see on the left-hand side, it says plus water, which means that that pot is going to receive water. And on the right side, it says negative water, which means it's not going to receive water. Looking at the center image, you see that your experimental group is actually going to receive the water versus the control group, which you're using as comparison, is not receiving anything. So fast forwarding to the end of the experiment, you're going to see on the left-hand side, the group that did get water, 9 out of 10 of those seeds sprouted, versus the right-hand pot that was literally in the same conditions, same soil, same seeds, same amount of sunlight and everything, didn't grow anything. Okay, I want you to notice something on the top. It says independent variable amount of water, dependent variable fraction of the seeds that sprouted. So I want you to actually try, and I want to see how much you actually remember from 6th and 7th grade science. I want to know if you can figure out the difference between the independent variable and the dependent variable. Take a moment, write down your answers, and we'll go over in just a moment. Let's see how you did. We're going to talk about the independent variable, or it can also be called the manipulated variable. The reason why it's called this is because the word manipulate means to change. This is what you're changing in your experiment and seeing how it affects your experiment. Basically, this is the only difference between your experimental group and your control group. There should only be one variable that is being tested. So if I go back to my example with the plants, let's stick with the amount of water. I'm going to choose a specific amount of water to be my control level. Let's say it was 1 cup of water per day. Anything that I change, let's say I have a second group and I do 1.5 cups of water per day, or I have a third group where I do half a cup of water each day. Even though I'm manipulating the amount of water, I end up having my control group at 1 cup and 2 experimental groups, one at half a cup of water a day and the other one at 1.5 cups. Moving forward, your dependent variable, I want you to realize that dependent starts with D. This is what you're collecting your data, okay? What type of data you're collecting. So this is what you're actually measuring. So I'm going to measure the growth of the plant based on the water and see how that comes out. Lastly, your controlled variables. There should only be one difference between your control group and your experimental group. Everything else is the same. So looking at your example here, the type of pot, the amount of seeds, the amount of soil, the windowsill it's sitting on, the amount of sunlight, everything was kept the same. The only difference in that example was that one group got water and one group didn't get water. The reason why you need to do this is so that you know that that one difference is the cause of all of your changes, all of your data being completely different. And data is extremely important. It's all the information you're gathering during your experiment, usually consists of a type of measurement. Okay, something with numbers, tallying, taking polls, having a bunch of surveys answered, and you recording all that information. Normally, you're going to organize these very nicely into graphs, charts, data tables, or diagrams, something easy to see, easy to view, easy to understand, quick views. Take a moment, answer the question that you actually see here. How can you make an experiment more reliable and valid? Hopefully you answered. We're going to go over your answers, and hopefully you answered that the more trials you have, the more accurate information you're going to get. The bigger the sample size, the more people you test, or the more subjects that you have to gather data from, the more valid your results are going to be. You're going to have a lot more to work with, a lot more to support whatever statement or whatever prediction you made earlier on. Don't forget, you can also go against your original hypothesis. Any information is good information. The more sample size and the more trials you actually do, your results are going to become more consistent. It reduces the probability of error, especially human error, because we are not perfect. After you obtain your results, you're going to have to make a conclusion. So in order to make a valid conclusion, or other words, acceptable or justifiable, you should begin by examining and analyzing all of the data you collected. Make a decision for the outcome of your experiment. So you're basically taking your data, ripping it apart, saying it supported my hypothesis or it did not support my hypothesis. Notice how I'm not saying proven or did not prove. Those are very strong words. Your conclusion must be supported with data. In order to write a very, very good conclusion, you should remember rerun. Recall all the information you collected, explain the purpose of the study, state your results, describe any uncertainties that exist, and state your next steps. What is something you would change for next time to make your research better or more reliable? Some things that I know none of you will ever do are some bad experimental practices, which means that you didn't test enough sample groups, or your sample size was too small, too few trials, your test didn't even contain a control group, which is definitely bad because you don't have anything to compare to. Your experiment cannot be recreated. That is a big problem. And additionally, your results were not significantly significant. On the next slide, I have a long paragraph. It's a state question. I just want you guys to try this, and the multiple choice answers will come right after. A student is studying the effect of temperature on the hydrolytic action of the enzyme gastric protease, which is contained in gastric fluid. An investigation is set up using 5 identical test tubes, each with 40 milliliters of gastric fluid and 20 millimeters of glass tubing filled with cooked egg white. The 5 test tubes are each placed in the different temperature-controlled environments of 0, 10, 20, 30, and 40 degrees Celsius. After 48 hours, the amount of egg white hydrolyzed in each tube is measured, which is a variable in this investigation. Your 4 choices are gastric fluid, length of the glass tubing, temperature, and time. Please take a moment, answer the question, and then I'll go over it. Hopefully you actually chose temperature as your answer. Now, if you're wondering why, let's think about it. Gastric fluid, length of glass tubing, and time were all kept actually the same throughout the whole experiment. Going back to the experiment, you could actually see the different temperatures controlled in the environment with 0, 10, 20, and 30 degrees Celsius. Whereas the amount each had 40 milliliters of gastric fluid, all had 20 millimeters of glass tubing filled with cooked egg, so that was all exactly the same. The only difference was the temperature. I hope this lesson actually helped everyone, and I hope you all study